Dear learners, I am Ramya, working as Assistant Professor in the Department of Biotechnology, St. Joseph's College of Arts and Science for Women, Hosur. Today, we are going to see the topic in microbiology bacterial appendages. What is bacterial appendages? Many bacterial cells have protein appendages embedded within their cell envelopes that extended outwards, allowing interaction with the environment. These appendages can attach to the other surface, which helps in transfer DNA or provide movement. Filamentous appendages includes mainly three, that is fimbriae, pillae, and flagella. Fimbriae and pillae. Fimbriae and pillae are structurally similar, and because differentiation between the two is problematic, these terms are often used interchangeably. The term fimbriae common refers to the bristle-like proteins projecting from cell surface by the hundreds. Fimbriae enables a cell to attach to surface and to other cells. For pathogenic bacteria, adherence to host cells is important for colonization, infectivity, and virulence. Adherence to surface is also important in biofilm formation. The term pili are called as longer, less numerous protein appendages. A specific type of pilus called as F pilus or sex pilus is important in transfer of DNA between bacterial cells, which occurs between members of same generation. Before the structure and functions of various components of bacterial cell envelope, scientists were already using cell envelope characteristics to classify bacteria. Various beta hemolytic strains of Streptococcus species using an agglutination assay. A technique uses the clumping of bacteria to detect specific cell surface antigens. Filamentous appendages, which is called as pillae and fimbriae. This is the diagram of the appendages, that is the outcome of protein appendages. Lansfield, a scientist, discovered that one group of Streptococcus pyogenes found in group A was associated with variety of human diseases based on variation in specific cell surface proteins, that is named as M proteins. Today, more than 80 different groups of strep A proteins have been identified based on M proteins. Various strains of group A streptococcus are associated with wide variety of human infections, including streptococcal pharyngitis, toxic shock syndrome, scarlet fever, rheumatic fever, and necrotizing fasciitis. The M protein is an important virulence factor which evades the immune system. Change in M protein alters the infectivity of particular strain of group A streptococcus. Is about capsule. Most prokaryotic cells have cell walls. Some may have additional cell envelope, which is extended from the cell wall, such as glycocalyx and S layers. The glycocalyx is a sugar coat, of which are of two important types: capsules and slime layers. A capsule is organized layer located outside of the cell wall and usually composed of polysaccharides or proteins. Slime layer is less tightly organized layer that is only loosely attached to the cell wall and can be more easily removed. It is composed of polysaccharides. Glycocalysis allows cells to adhere to surfaces, aiding the information of biofilm. In nature, most microbes live in mixed communities within biofilms, partly because the biofilm affords some level of protection. This is the diagram of capsule. The ability to produce a capsule can contribute to microbes pathogenicity, that is microorganisms can ability to cause the disease because the capsule can make it more difficult for phagocytic cells to engulf and kill the microorganisms. Streptococcus pneumoniae, for example, produces a capsule that is known to increase its virulence. Capsules are difficult to stain for microscopy. Negative staining techniques are typically used. Flagella. Flagella are structures used by cells to move in aqueous environment. Bacterial flagella rotate rather than move side by side. They are composed of protein called flagellin and extend outward from the cell's membrane. Different types of motile bacteria exhibits different arrangements of flagella. A bacterium with a singular flagellum, typically located at one end of the cell, is known as monotrichous. Cells with amphitrichous flagella have flagellums or tuft of flagella at both the ends. Example is Spirillum minor. Cells with lophotrichous flagella have tuft of end at one end of the cell. Example Pseudomonas aeruginosa. Flagella that covers the entire surface of bacterial cell are called as peritrichous. Example E. coli. This is the structure of flagellum: monotrichous, amphitrichous, lophotrichous and peritrichous. Directional movement depends on the configuration of flagella. How the flagella movement will take place in bacteria? Bacteria can move in response to a variety of environmental signals including light, magnetic fields, chemical gradients. Purposeful movement towards a chemical attractant like a food source or away from a repellent like a poisonous chemical is achieved by increasing the length of runs and decreasing the length of tumbles. The movement of flagella will be two. One is tumbling movement, clockwise rotation of flagella, and another one is running movement, counterclockwise rotation of flagella. Bacteria achieves directional movement by changing the rotation of their flagella. In cells with peritrichous flagella, the flagella bundle when they rotate in counterclockwise direction, resulting in a run. However, when the flagella rotates in a clockwise direction, the flagella are no longer bundled, resulting in tumbles. Today's question is, who discovered phagocytosis process? Thank you, dear learners.